Great, thanks. So I would like to introduce our next keynote speaker. It is Ole Seeberg from Natural History Museum in Denmark. Ole is a full professor and head of the Botanic Garden at the Natural History Museum of Denmark, Copenhagen University, and got both PhD and Doctor of Science degree for University of Copenhagen. He has previously been head of the Botanic Institute and member of the board of the University of Copenhagen. He's trained as a classical botanist, but has been engaged in the molecular revolution from its beginning. He has been involved with various aspects of biobanking for the last 10 years and had a key role in establishing GGBN, which is absolutely true. So he was the chair of the interim executive committee, and tomorrow we will vote for the new chair, which will be probably the same. So Ole, the floor is yours.
Hello everybody. I just have to give you an announcement because Walter Berenson from the Botanical Garden asked me to tell you, if, in case you don't know it, and if you're interested, that on Saturday, June 25th at 1:45 p.m. at the Rousseau-Saal in the Botanical Garden, there is a meeting of the Global Plant Initiative, just that you know it. And you can probably contact Gabby if you need more information about it, but I promised Walter to give that information. I don't expect you to kill yourself during my presentation, as it was suggested by the previous keynote speaker. I would be fully satisfied if you don't like the speech, if you just fell asleep. We'll wake you afterwards. I'm going to tell you something about what GGBN is all about and what we're trying to do, preserving a cross-section of the tree of life for eternity, so to speak, during my talk. I'm going to build my talk on first giving you an indication of what is the size of the problem we are dealing with, if we're going to sort of sample most of it, and why it does matter, taking advantage of the tree of life, which is a key component in what we do, and then give you an indication of what GGBN are, built on an introduction, our data standard and access task force, our policies and practices task force, and the marketing and outreach task force, and then give you an overview of the current status. Let's first look at the size of the problem. Well, honestly, if I ask the question on the upper part of the slide, how many species are there on Earth, you'll probably be unable to answer it. At least I don't know, and honestly, nobody really knows. But if you look at these figures, these are sort of two different indications of how many species there are. The upper one is the latest one, and perhaps the most scientific one, made by Mora and al. And published in PLOS ONE, that indicates there is approximately 1.5 million known species and 11 million that we don't know, or estimated a total of 11 million. And we only know about 13% of those. The lower figure shows the classical estimates by Gombrich and Jenkins that has been out there for many years, which indicate 1.85 million species and 14 million. But the interesting thing is, no matter which of the figures are right, if you look at it, we only know 13%, no matter whether we take this estimate or this estimate. So there's a lot of things out there we don't know, and even collecting these 13% we already know is a formidable task. Probably I don't need to spend too much time of this, but as you traditionally know, or know sort of from reading, there are three levels of diversity. There's genetic diversity, which is about the interspecific variation, genetic variation, the species diversity or taxonomic diversity, and ecosystem diversity. And needless to say almost, GGBN has nothing to do with ecosystem diversity, but try to capture the other two types of diversity. The reasons for doing so are obvious. Most of you also probably are familiar with these different usefulness or the different usefulness of biodiversity all the ecosystem services that are not directly of our interest, but we are probably more centered about these more sort of direct uses, and if you ever discuss with anybody the usefulness of biodiversity, just ask them to go out and open your fridge, and it will be full of biodiversity. So there's no reason to believe that biodiversity is uninteresting, and of course it has a lot of social, educational, etc. Components. So, as I said in the short welcome introduction, I think that the future of biodiversity research is to a large extent linked on our ability to make genomic samples available for research and accessible for research. That's, to me, very important. And of course, we need to have this collaborative effort in order to do it. So why does it matter? Why does we need to have these things in storage? Well, it has been known for very, very long that biodiversity is of immense importance to humankind. And it's said in the slide that I just passed by pretty quickly. This goes far back in time. In the left-hand side of this figure, you can see one of the old herbal books by Leonard Fuchs, who was very well aware that these plants that he were actually looking at had a lot of medical properties, and he gave out a so-called herbal, and of course there were many herbals at that time, and it's still that's the same manner things are being used in certain parts of the globe. Here it is a Chinese herbal shop, and a herbalist sort of weighing out different herbs that can be used for remedying different diseases. And you don't have to go to China, you just have to go to Vienna. Here you can see this to the right-hand side, there is a herbal shop, and on the other side there is an apothecary. So it's still, well, I hope they're doing their different sort of task. This one is sort of the modern way of medical, and this is sort of a more traditional. But there are, of course, other sort of very, very important components, for instance, in plant. If you look at here at the Madagascar periwinkle, up there, you probably know that it includes two substances that are extremely potent in the treatment of leukemia, especially in children, and in treating Hodgkin's lymphoma. So we know that the stuff is out there, and we know that, of course, that it's genetically controlled. Alzheimer's disease and snowdrops goes finally together. Snowdrop includes a component called galactamine, and we also know that this stuff cannot stop Alzheimer because it's, well, we really do not know a lot about Alzheimer, but we do know that it's a medicine that can actually relieve some of the problems with Alzheimer, at least for a while. It's not a cure. But there are also other obvious uses. Here we have this interesting little frog, from which we have extracted a small component, a synthetic gene, dermateptide B1, that transferred to potatoes, genetically modified to potatoes, infer resistance against a broad range of bacterial and viral infections, including such things as dry rot, late blight, and pink rot. So there are many cool ideas in the genome. It's also well known that some morphine mimics found in cone snails are extremely good at controlling transmission of signals between nerve cells, and even in very, very small quantities, the concotoxin syncotide is very, very efficient at interrupting transmission, and it's a very, very good painkiller. So that's also an important thing. And sort of men of my age are pretty satisfied with the fact that from this pit viper, one has extracted ACE inhibitors, which can be used in medicine that control blood pressure and cognitive heart failure. I guess most men in my age suffers from too high blood pressure, so I'm pretty satisfied with this pit viper. But going beyond sort of traditional uses, there are also other uses. If you look at this shark, not only this shark, but sharks in general, it's known that even though they often digest a very, for us, very unhealthy meals. Bacterial infections in sharks are very rare. And the reason is that the surface of the skin of the shark has a very complicated structure that for some reason do not let the bacteria settle on them. And this information has already been used in fabrics that is being used in the hospitals to reduce the risk of transferring bacterial infections. And some of you will like this creature, the immortal jellyfish, the jellyfish that never dies. Usually these coelenterate goes between a polyp and a medusa, and the medusa is the fertile stadium which have sexual reproduction. But sometimes if this little creature finds it's too boring to be a medusa, well, let's transform myself into a lump of cells that drop down to the bottom, settle down, and create a new polyp. So this polyp again butts off a medusa, so in that way you can sort of live forever. And that's probably, well, I don't know if it's something you want to do, but at least it's an interesting way of sustaining life forever. So why does it matter we preserve this? Of course, it's important for us that we have these resources available for the kind of science that is being done now. It's important for human welfare, and it's important for our future. And with the very pessimistic sort of talk, or maybe not very pessimistic, but the requirements set for us by the previous speaker, I think it's important to realize that extinction is forever, and conservation might fail, and eventually the only thing we might have left is actually the samples available, for instance, in cryo, which is much better than if we only had a dried specimen. So then what can we do in order to get an optimal sample of the tree of life? Well, first of all, I guess we are the first generation that has a really solid grip on the tree of life. Previous ideas were much more vague. It was actually Charles Darwin that made the groundbreaking world for having the tree of life, but he himself never produced anything that could be called a phylogeny. He had a theoretical tree in his Origin of Species, and besides that, nobody sort of thought he was ever thinking about phylogeny, but he was. When he left the Galapagos Island in 1836, on his way to Tahiti, I think it was, he sat in his cabin and scribbled the most famous, well, cartoon almost, the most famous tree in the world, I think, talking about the relationship of birds on the Galapagos Island. So that's the first real phylogeny that was produced. And of course, now if you go to a modern phylogeny, you can see that man also have a position. This is a phylogeny of all of life based on molecular data, and man also fits in somewhere up there, I think. So how can we use this information? How can we use the information of the tree of life to do an intelligent sample for eternity? Well, we can actually start on the left-hand side and look at the taxonomic hierarchy and realize that there are 3 domains, 64 phyla. Of course, I know this can be discussed. There can be 5 domains and there can be 73 divisions, but the order of magnitude is probably correct. And if there are 14 million species at the end, this is sort of the scale of at least an important part of the hierarchy of life. And if we have a phylogenetic tree like this, we can decide that, well, we're going to do a very fine sampling of the tree. We are actually going to take an exemplar of everything to the right of this line. This would indicate, in this particular case, that we will have to sample each and every species that exists. For this little tree, this will of course be possible, but it will probably not be possible if we have 14 million species to deal with. So if we aim at a slightly sort of medium dense selection of the tree, we would take just one of each of the clade that are cut off by this group. or by this line, and are to the right of the figure. So we'll have a much smaller representation, but we will still cover a large part of the tree of life. Of course, we can go to a very coarse and very small sampling, just take one exemplar of any clade to the right of this figure or this line. So that's a way we could actually sample the tree of life without necessarily have to do it all. And why is this permissible? Well, if you look at this figure, which is based on vertebrates, mostly on mammals, it's common knowledge that 98% of the genetic makeup of a chimpanzee and Homo sapiens is more or less the same. So preserving Homo sapiens and a chimpanzee wouldn't give you much extra information. And it is surprising, perhaps, that if you look at the shape of this line, or rough line, still, if you go all the way down to Xenopus, about 60% of all the genes are the same as are found in the chimpanzee and Homo. So if you sample Homo and a couple of points there, you actually have an enormous amount of the variation that is out there. And you could even say you had a lot of superfluous information, but you probably don't. So we can actually, by sampling the tree of life intelligently enough, we can sample an enormous amount of genetic variation that is dispersed across the tree. Of course, the dream in certain instances, for instance, agriculture, would be to capture all the genetic variation there is out there. But you can easily imagine with 14 million species, and if we have to catch, say, 500 of each to catch about just a part of the genetic variation, the task becomes even much more formidable than collecting just the 14 million. So why do we need this network? Well, we need this network because a lot of scientists say that one of the biggest bottlenecks for doing research is the lack of samples. There's no way you can get a quick assessment of availability of samples prior to project start. The bottleneck is very easily seen. So we need, as a scientists that are doing this work, they need to identify gaps in our sampling. They need to know what we have out there, and we need a guideline for collecting what we do not have. It's pretty obvious, but at this stage, we are the first that really can identify these gaps. And I'll show this slide again, because I think one of the big things for the future is to do this translational museomics, as we can call it. And it's, as I said before, it's an interdisciplinary branch of biology that supports the integration of natural history collection, modern laboratory methodology, and societal needs. This doesn't mean that I know that a lot of current sort of genomics just go into the collections, use them, and provide nothing back. So that is not what I consider interdisciplinary and integral sort of way of dealing with the collection. So it's got to be a much more united effort to actually do genomics on the museum collection and still pay something back to the collections. What then is GGBN? Well, our primary idea is to make genomic collections discoverable for research through a network community of biodiversity repositories. And the vision is to make a global network of well-managed collections of genome-quality tissue samples from across the tree of life for the benefit of society through biodiversity research, development, and conservation. And we want to foster collaboration among the repositories of biological biodiversity to ensure quality standards, because these are very important that we are dealing with the samples in the same manner, improve best practices, secure that the databases are interoperable, and harmonize exchange of material in accordance with national and international legislation and convention. And that is, of course, going to be an increasing problem or an increasing challenge, because we, as at least most of the audience the audience here is representing, well, the Western world with quite a lot of money compared to other parts in the globe, and we have to foster trust in the biodiversity-rich countries Otherwise, we are never going to succeed in sampling the tree of life. So the overall goal is, as I said before, provide genome quality samples for across the tree of life for research, training and development, and contribute to conservation of global genetic diversity for the future. And I'll sort of just briefly take you through the long term or the long version of our goals. So we intend to provide access to a global data management system hosting the aggregate primary species data and associated metadata for all member institutions. That's a work that is already far progressed, and if you ever have gone to our website, you'll easily see what you can find there. And of course, we need to have a common standard for sharing tissue and DNA information, otherwise we cannot talk with the database. So we also have developed a best practice related to management and stewardship of genomic samples and their derivatives, and have created access and benefit sharing documents that are already available online. And we are encouraging target sampling and preservation of genomes representing a synoptic sample of the life on Earth. And then, of course, we have to create and to recruit new partners, and both on a regional basis and taxonomic focus to help reach the goal. And we have to emphasize that GGBN is not centered on non-human diversity. There's a lot of institutions out there doing human diversity. All the medical world is doing that and has been doing that 10 or 15 years before we ever started. So that's not our goal. So everything was started in 2011 at a meeting in Washington where 13 European institutions, or 13 institutions representing seven countries were present, and we decided simply to go on creating GGBN. It was done very simple. We created a steering committee was supposed to oversee the work, and we had a series of task forces that did the actual work and reported it back to the steering committee. As of this morning, we have changed our structure. We are no longer an interim steering committee. The General Assembly has devised a new executive committee, and we have created a secretariat which is placed in Washington, and a technical management system that is placed here in Berlin. And we will take a closer look and see if we shouldn't have more task forces. So what has the Data Standard and Access Task Force group done? Well, actually, everything sort of takes its outset in GBIF, and that's why I show this picture from GBIF, where the primary species data are stored, and this is just a simple case showing the distribution of a group of hummingbirds, and even in this clean data set, you can still see that there are a few outliers. There are no free-flying hummingbirds in Europe, as far as I know, but still there are, they live only very shortly at least. So that is what is the primary entry point to most of our data, that is the specimen data and GBIF's species records. But one of the things that we've had to deal with is these very sort of complicated things that you don't pay much attention to if you haven't tried it. This is sort of the classical pipeline that I've also showed this morning. That either you find a seal or you put it in your collection if you have space enough. Then you have a real wildshot, not even just a photo. You take a blood sample, you do a genomic sample, and you have a few sequences and from one species. That's easy to deal with. It can easily be handled. That's what we do traditionally. But there are other things. This is another option. You have no voucher, but you find in this particular case, a fecal sample from a seal. You extract environmental DNA, and you have thousands and thousands of sequences from dozens of species. And that starts to create problem because we are used to having a system where we can just type a species name and get out a sequence. That is not possible in the same sense here. There is no specimen to link it to. And another situation is ancient bones, where you have ancient DNA, where you have a few fragmented sequence for just one species. So there are several things that we have to take care of when building this database, and I would say just currently this works perfect, and this is more or less work in progress. We have had a workshop early, before this meeting started at all. How do we deal with an environmental sample? What should be the entry point? Should it be 200 milliliters of water from somewhere, or should it be somewhere else, something else? And we have to, as I said before, this is not about humans. There are, however, exceptions, because as you all know, humans are attacked by various insects, and we have certain unwanted visitors, and we are pretty bad. The natural history museums are pretty bad at storing this type of information, and GBIF is probably not very well suited actually to convey this information. And finally, there is a problem of ancient human, which quite a few natural history museums have, where we have difficulties because the management systems are often separate, and GBIF does not really connect well with this. So we gotta develop data standard and community conventions to really be able to handle this data, because we have some of these data. As I said before, this is not about humans. It's not about deep freezing human bodies as they do in the Cryonics Institute and reviving them when times get better or the illness can be remedied. But there are, as I said, there are exceptions like the snowman or the iceman Ötzi that was found in the Alps in 1991. He could easily end up in a museum environment, and we would have to be able to deal with this. So there are problems with our different types of samples and DNA that goes into the samples. There are taxon information that can be based on a voucher in a traditional way, or it could be based on an environmental DNA. And of course, we have to create conventions that can deal with all these things, because it would not be a good idea if a scientist looking for seal DNA got a lot of environmental samples. So it's a challenge that is constantly being worked on, both by us and by GBIF, and of course in collaboration with GBIF. This just jump over. So what we want to do is to be able to link information between sources, material, the analysis of data, and we are sort of sliding in this area where we can actually link all this type of information from GBIF to the data and actually out to the publications that are available. So just a brief overview. This is of course based on the GBIF pipeline, and most of you, or many of you have probably heard about the ABCD and Darwin Core standards, and I won't say much about this, but if you're very interested, you can go to this publication, which is actually a description of the data portal. So what has been a major problem as well for both the GBIF and us has been that a lot of the information provided out here are not constantly available, because sometimes, well, in the best of all worlds, the data providers are up and running all the time, 24 hours a day, but that's not the reality. A lot of providers goes off and on and off in different times. So the Berlin group has devised a system that actually store a clean subset of these data, and these are actually the ones you search when you go into our data portal and get your needed information out. So here is a couple of examples of the database as it is, where you can actually switch between DNA and taxon information down here and down there. So if you, for instance, let's switch between the taxon name and the tissues, and if you go in here, for instance, take a quick look at this little fish, what you'll get back is something like this, that there are approximately 10,000 records in GBIF. There are 219 nucleotide sequences. There are 63 barcode sequences. The fish have a species page in Encyclopedia of Life, and the GGBN has 22 DNA samples and 47 tissue samples. That's the kind of information it would take you a relatively long time to get in any other manner. And what you can then do, of course, is request these DNA from or tissue through our portal, download all the sample information, and you have to go to the member in order to get access to the sample, because it's the institution itself that is responsible for all further steps, checking availability, and not least that they are compliant with the ABS system, and the loaning conditions are okay. You might be able to, or you might have to pay for it if you're going to pay maybe post it and package, and you may be required to sign a material transfer agreement, and then they'll have to ship you the samples. That's sort of the technical setup, but up till now I don't think we have sold any samples or provided any samples yet, but we are increasing very rapidly, so it will soon be a reality. And of course there are guidelines on how to actually acknowledge the provider of the samples. Policies and practice, of course, has to be a standard way of dealing with these things, and in that respect we have had a work group that have been looking at best practice recommendations, and we are standing on the shoulder of giants in some respects, because the medical biorepositories has already made best practice guidelines for biorepositories that deal with human samples, and in the third edition we had an input on the necessary changes in this document to deal with biodiversity, and in the next version that comes out hopefully in one and a half year, we will actually have a massive input of what is needed. And the rationale between dealing with ISPER instead of developing our own document is there's no reason to repeat what is already out there, and a lot of the information has already been provided by the medical world. So we just pick up and enlarge it, actually with consent of ISPER that we are closely affiliated with. We're also trying to build a document library that will keep all information in one place, so you can actually search for, best practices other than our own. You can search for literature where the samples are cited in, but that's a work in progress that we haven't made yet. One thing we have had to spend an enormous amount of time on is, of course, ABS compliance. You all know the problems related to the Nagoya Protocol, and we have again decided not to try to do our own stuff. So what we have done has been done in very, very close collaboration with CETAF. So the CETAF documents and our documents on ABS are perfectly aligned and more or less the same, except the headlines. So that's, and again, as I said, it's very important to raise transparency and traceability of where the species are used, and eventually we have to put that kind of information into the ABS clearinghouse when it gets up and fully running. So this is more or less the same again. So where are we now? Well, I think we have to go just give you a short overview of what the marketing and outreach group has done, and they have sort of identified who are the key users or potential users of GGBN. And this has been done both on interviews or on surveys. And the interesting thing is that just now, most of the members of GGBN are all natural history museums. But we are reaching out to other communities like zoos, not botanical gardens, zoos, aquaria, crop storage facilities, seed banks, etc., because the plan is to include all of them. So these are what people consider the most important things, being members of GGBN. That makes their collections discoverable, and they are a member of a community that actually creates standards for the genomic collections, and they provide biorepository contributors with a community standard and best practice for collection of data and management of genomic samples, and then they provide contributors with access to communities that provide storage facilities for and access to information on their own genomic samples. So we are sort of trying to align ourselves with this. And again, the most important thing is to have trusted and transparent access. Otherwise, we run into problems with the providers. So the current status of GGBN is that we are 48 member organization worldwide. There are huge chunks of the globe where there are very few. Africa, South America could be better covered, even North America. And of course, we have very few members here. And we have a huge concentration of members in Europe, for primarily, not exclusively, but primarily due to the synthesis project. So we have a number of GGBN members, increase in the number of members since we started in 2011, close to 50, and the number of collections online is probably about 17. Okay, 17. So it's increasing pretty quickly. And the number of samples are close to 500,000 from a very, very modest start. Of course, the number of families is much bigger than the number of genera or the number of species, but we are constantly increasing and hope to do that more. This is an overview of what happened, where the participants in the first Congress came from, here given in purple. And thank you very much for listening. These are the people that have supported this work, institutions, etc. So if there's any question, just feel free to ask.
Hello.
Hello.
I work in an entomological collection.
Yeah.
I don't know a lot about modern techniques to extract DNA. And my question is, can you extract DNA from old specimen? And if you can, do you have to destroy the old specimen? I mean, for example, a small beetle.
No. You can. Well, sometimes you can just pull off a leg, and then extract the DNA. And other times you can actually extract the DNA and almost let the exoskeleton be there on its own. So it's absolutely doable, but it's of course, the more degraded the DNA, the more difficult it becomes to assemble larger chunks of DNA. So it depends on your goal. If you only want a single sequence and you have a relatively fresh museum specimen, you can do it. But if you want to do the whole genome, it's probably very expensive to do it. Everything in life is much easier if you have a fresh sample.
Hello.
Yeah.
Wolf Eiserhardt, Kew Gardens. Thanks for a really nice talk. You suggested in the beginning that the genomes of humans and chimps are so similar that it's maybe not worth bothering with both. Isn't that the same thing as saying we're not really interested in the information that makes them different? And is the preservation of closely related things not maybe in some cases just as important as the preservation of distantly related?
Yeah, but of course it can even be more interesting. But given you are completely ignorant, you can use this model as a sampling system. But because there will be many places where we are completely ignorant of what are the importance of these species. Needless to say, the agricultural community has a very dense sampling of a lot of wild species, and this is of course completely legitimate. It's not an attempt to say we shall not collect as much as we can. It's just a way of indicating that if we have no other knowledge, we can use the tree of life to actually sample in an intelligent manner. Of course, we will both need gorillas and chimpanzees and orangutans, etc., etc. There's no doubt about that. It's just a way of dealing with the immense amount of biodiversity. We have absolutely no clue about any importance of except that they're there.
I completely agree. Thanks a lot.
Hello, Dennis.
This is sort of a comment, but it's also a question. Lately, some of the people, particularly like at Coldstream Harbor, where I do all my sequencing, are going, it costs too much to store this information. We're going to dump it and we'll resequence it as the price comes down. That brings us to the problem of when you get some weird little plant, in my case, and you have a limited sample, of using up the sample because you get all of these requests. You have any notions about that coming along?
Well, I agree that it costs money to store samples. It costs money to store data, and it's a trade-off, I guess. For certain species, you only get this one sample forever, and it's not more different than any other things in a natural history environment or in collection environment. You have the number of samples you have, and in each and every case, you have to make a judgment. Can I salvage this sample for this investigation? Is it important enough?
Well, I mean, you will run out eventually if you don't
No matter what
collect it, or
yeah,
if it's extinct, you're not going to get it.
But if you have, say, 10,000 samples and you got to be able to argue for the fact that you're going to take the last of this. But that goes for everything.
Ole, perhaps this is something you were going to do. It just occurred to me, I'm John Coddington, I'm on the Interim Executive Committee, and I thought it might be good for GGBN to ask the SPNHC membership a question that might go like this. Are you work for a collection or associated with a collection which has, is either thinking about growing a cryo collection, has cryo material in freezers, are facing issues in collection management of that, and are intrigued with the possibility of joining GGBN? If you would raise your hands, in other words. Only 3, 4.
No, there's a lot behind you.
Well, talk to us.
This was a PR stunt. Okay, thank you.
Thanks, Ole, for this talk. And before everybody is leaving, those of you who haven't paid for the pub quiz, please do it now. And we will leave at 7:30 here. And what? Yeah, meeting in the foyer. And everybody who is in one of the task forces of GGBN, the work is not done for today. I'm sorry. We will